Okay, so we are moving on to our next unit, Unit 2. This is cell structure and function. Obviously, cell. Well, what is a cell? This is kind of the smallest unit of life. Smallest unit of life. Um, so this is going to be what our next topic covers. Everything from why cells are small, all the way to how they function. Some of the membrane-bound organelles that have very specific tasks inside the cell. And very specifically water potential, which is kind of the main topics we're going to talk about. So, first of all, as you guys probably already know or remember from uh, biology class, you have eukaryotic cells and prokaryotic cells. Eukaryotic cells have these things. Eukaryotic cells have membrane-bound organelles. Membrane-bound organelles. These are structures within the cell that compartmentalize the cell and let it do many different things. Prokaryotic cells are much smaller. We'll talk about these later on. We'll talk about these differences more in a more detailed fashion later on. These are smaller, and they do not know membrane bound. They don't have that. They do have ribosomes, but they don't. But the things that are bound with a membrane, such as a nucleus or a endoplasmic reticulum, as we're going to talk about, these don't have. Prokaryotic cells and eukaryotic cells both do share this first feature I want to talk about, which is the cell membrane. The cell membrane is the outer boundary of a cell, an animal cell particularly. Plant cells also have a cell wall, and they regulate what enters and leaves the cell. What enters and leaves the cell. This allows stuff in, allows stuff out. As you know, as we talked about in our lipids video, this cell membrane is made up of these things called, draw some of them down here. Cell membrane is made up of these things called phospholipids, right? Phosphate groups attached to these hydrophobic tails here, right? But there's all sorts of stuff going on in the membrane. There's proteins embedded in here. There are glycoproteins with little sugars coming off the end. There's all sorts of things in this cell membrane, but just keep in mind the basic structure is a phospholipid bilayer. So mostly hydrophobic portions and hydrophilic portions, and it surrounds the cell. It is what keeps the stuff inside, and what keeps the stuff outside. So, first organelle we're going to talk about is the, and I hesitate to say most important because all the organelles are very important, but this is where our DNA is contained and segregated from the rest of the cell. Again, this is a membrane-bound organelle, meaning that there is a membrane outside of this nucleus, right? A phospholipid bilayer as well, kind of keeping, it has these pores in it, as we're going to talk about. But this is a membrane-bound organelle, meaning that it has a membrane. And inside of the nucleus, as you might guess, is our DNA, usually bound up, wound up, stuffed into these chromosomes, especially during mitosis when you have these cells need to divide and this nucleus needs to divide. But in general, this is where our DNA is stored, A, T, C, G, all that good stuff, the stuff that makes you is stored here in the nucleus. So, ribosomes, these are not a membranous organelle. These are made up of RNA and proteins. So ribosomes are made of RNA and some proteins. Very, very useful organelle here because it is what actually transcribes or translates our RNAs coming from the nucleus. Right? You have these. You probably remember this as well, but mRNAs, right? They kind of flow out of this nucleus. This is our messenger RNA. Our messenger RNA gets translated into protein at these ribosomes. Which are these red things. They exist kind of all throughout the cell, generally found very close to the nucleus, and they spit out these proteins, right? These long chains of amino acids. Again, this is hugely polypeptide. Peptide comes out the other side, right? So DNA to mRNA to the ribosome where it gets translated into protein. This is something we'll talk about much, much more in depth later on in the year, so don't get too caught up in it. But I would like you to know this basic process and the basic function of a ribosome. It is where proteins are synthesized. Okay. Our rough endoplasmic reticulum, very big fancy word. This is a membrane-bound organelle. It's also membranous, meaning it has these phospholipids throughout here. And you also have ribosomes that are studded within this rough endoplasmic reticulum, hence the word rough, right? Because it is kind of has this studded, these ribosomes kind of poking out everywhere along this membrane, right? This is the rough endoplasmic reticulum. A lot of protein synthesis takes place here. It is usually continuous with the nucleus right here. If you'll notice, this membrane kind of turns into, nuclear membrane kind of turns into the rough ER, where you'll find a bunch of ribosomes throughout here making proteins, doing what it needs to do, right? These proteins are, again, this DNA is everything, right? This is what codes for exactly what you and I are, and every organism. Right? Okay, smooth endoplasmic reticulum. This is something that's also kind of continuous with this layer. It has very specialized function in certain types of cells, such as muscles and things like that. But this typically synthesizes lipids and also stores ions. So lipid synthesis, this is where lipids are made, and this is where ions, such as calcium, the calcium is stored in this. This is very important for muscle cells in your body. Ion storage occurs here. You'll notice that this is smooth endoplasmic reticulum. It is continuous kind of with the rough endoplasmic reticulum. It's connected, but it does not have all these ribosomes in it, so it is not rough. This is smooth endoplasmic reticulum. Okay, the Golgi apparatus. Again, you've probably seen this and heard this described as the flat stack of pancakes. This helps to package proteins, typically for export. So once these proteins are made, they typically enter these little vesicles. That little down vesicle is just kind of a membranous sac, right? And it enters one side of this Golgi body, and it goes through here. It's processed, is folded as it needs to be folded in many cases, and then typically is maybe exported in the cell, right? So this basic process, again, I'll go over it again. You have DNA, right? Transcribed onto mRNA, which leaves the nucleus here. Gets picked up probably by a ribosome. Protein gets translated. This protein gets picked up by a vesicle. And this protein is processed, folded, and basically shipped. Some people have heard of Golgi apparatus, the cell, because it kind of determines where stuff is going. And it also, keep in mind, also helps to fold these proteins specifically the way they need to be folded. So, the Golgi apparatus. All right, everybody's favorite, mitochondria. This is where the ATP is made. This is where the energy, or that we use is generated. This chemical energy. Keep in mind, this is was evolved. We have kind of co-evolved with this mitochondria. This mitochondria was engulfed by eukaryotic cell a long, long time ago. And, many, many, many years later, we have this kind of relationship where we can't live without them, they can't live without us. They generate the chemical energy of our cells. Sugar goes in typically, and energy comes out. How we generate energy in the body through a process called, I already wrote it down here, but cellular respiration. Which, again, we'll talk about that much more in depth later on, so you don't need to know the details of it. Just know that this is where energy is produced. Okay, some cells, I'm sure you know which kind. You've seen these before, but some cells have these things called chloroplasts, right? These are typically found in autotrophs, right? Which, if you remember from the ecology unit, is the word for something that can generate its own energy from sunlight. So sunlight comes in, these photons come in, right, and kind of strike these chlorophyll molecules contained here in these membranes in the chloroplast, and you have some energy is produced, but you have some sugar comes out, right? This is stored as sugar. Of course, plant cells also have these mitochondria, and what do you think that mitochondria does with the sugar? It gives energy to the rest of the cell for all of these processes that you kind of see going on here, all these generation of proteins, Golgi body. All of this comes from ultimately these chloroplasts fixing this photon energy into a chemical form, which is sugar, which the mitochondria then uses. So that's the chloroplast. That's the site of the process called. In contrast to cellular respiration, photosynthesis. That's something you've heard about before, I'm sure. Photosynthesis. Again, this is something we'll talk about very in depth in a later unit. It is where solar energy is converted into chemical energy in the form of carbohydrates. Just something to keep in mind. Um, Photosynthesis occurs in the chloroplast. Cellular respiration occurs in the mitochondria. Also important to keep in mind that the chloroplast is another organelle that co-evolved, that was engulfed by a cell a long time ago. And now these autotrophs cannot live without these chloroplasts. The cells cannot live without, or the chloroplasts cannot live without these cells. Prokaryote has its own DNA, just like mitochondria. Okay, vacuole. This is an enclosed cellular compartment that contains inorganic and organic molecules depending on the cell. It is very important in plants because it kind of helps to store water, helps to maintain water levels in plants. As plants take in water, they need a way to control the input and output of water. Typically, they're stored in these vacuoles. Also, and animal cells have these as well. They're just kind of generalized structures with many purposes. But they store many useful proteins and sometimes ions and molecules that need to be saved for other cellular processes. So it's kind of like a storage depot of the cell. And finally, the last one I'm going to talk about are lysosomes. These are small vesicles. Again, you see that word, right? Vesicles are just these little membranes. They contain digestive enzymes. Digestive enzymes are contained in these lysosomes. These help to break down stuff in the cell. It shouldn't be there. Or break down stuff that is dead. You have a mitochondria that is no longer functioning. A lysosome will eventually engulf it and break it down, digest it, and release its component parts back into the cell. Right. So these lysosomes are very important for the recycling of materials, of cellular material. They also help in apoptosis. So, programmed cell death. I'll write that word over here. Apoptosis. That's probably the word you've heard. But sometimes when the cell needs to die and there's something wrong with it, it just needs to destroy the whole thing. These lysosomes will release these digestive enzymes, and they'll basically eat through everything in the cell, destroy it. So lysosomes, kind of like the garbage men of the cell, very important. Again, you see this word. You've probably heard the word hydrolysis, to break down. Hydro. I'm just going to remind you because there's a word here that might help you remember. Hydrolysis. Lyse, to break down. Lysosome, this is to break down. So this, something to keep in mind. Maybe that'll help you remember. The lysosomes break stuff down in the cell. Okay, that's it for topic 2.1. I hope that was helpful, and I hope you drew all of this on your own paper. That way you can remember it. All right.